Great. So can y'all see that? We're good to go? I'm presenting today work that I completed while I was a postdoctoral fellow in the Martin lab at Bucknell between 2017 and 2019. And what I'll be talking about today is a project, one of the first projects that we got started on in the Martin lab concerning population genetics and species of concern in Pennsylvania. And I'll be telling you about Erigenia bulbosa, which is in the Apiaceae, which is an edge of range species, which we've heard a lot about today from the great talks by Cheyenne and John. So this project, as well as the previous two you've heard about, is the result of a partnership between conservation practitioners and academic institution. Namely, that's the Western Pennsylvania Conservancy and the Natural Heritage Program, as well as Bucknell University. And this is through some funding that was obtained through the Pennsylvania DCNR Wild Resource Conservation Program that Dr. Chris Martin and Dr. Scott Schutte had applied for and was successful. And you've already heard about Bactesia and Casmanthium. I'll be telling you about Erigenia, and I'm sure that they'll have more great projects coming for you all in the future. So I want to start with telling you a little bit about the biology of Erigenia bulbosa. Erigenia is super interesting because it's a monotypic genus. So it's the only species in its genus, but that doesn't mean it's lonely. It's pretty cool. It's in the Apiaceae, as I said, and it's the harbinger of spring because it's one of the first plants in the spring that comes out. It blooms between February and April in mesic-rich, well-drained woodlands and forests. One of the common names for this plant is pepper and salt, and that's because it's got these really awesome, can you see my mouse? Yeah. Maroon anthers and white corollas that kind of give it this like speckled appearance from above. It grows vegetatively for a long time from small tubers. We suspect this occurs between 5 and 6 years, and the first flowers don't actually appear until its 6th or 7th year as a living organism. As I mentioned, these are spring ephemerals. We know they're herbaceous and they're long-lived, and that's kind of what we know. There's not been a lot of work or research done on this plant. We suspect that it's an outcrosser and that it's likely, visited by bees and flies. And I've also found some literature that specifies that the anthers and the ovules mature within the same plant at different times. And so this would promote outcrossing between individuals within the same population. The fruits are capsules that kind of just, they don't explode or anything. They just kind of fall apart and fall to the seeds can fall to the ground. And the species is not clonal, so it does not produce clones. And it's also diploid, which makes it really great for population genetics work. So the distribution in North America, this plant occurs from between the Oklahoma, Kansas area, or the central Great Plains, all the way over to Pennsylvania and New York. We can ignore these New York populations and just say that Pennsylvania is the furthest east. I'm just kidding. But anyway, it also occurs between Wisconsin and Alabama. In Pennsylvania, within Pennsylvania, there is a disjunction. So there's a gap here. And some of these, this does include some historical points as well. So I'm going to zoom in now on Pennsylvania and talk about that. So within Pennsylvania, I have marked on this map the modern sites, plants that have been found since, or populations that have been found since 1992, as well as some historic sites, and those are denoted with X's. And those have not been recovered in multiple surveys since the mid-90s. So we know we've got some likely loss of some historical populations, and that could be due to land use changes, but also it could be due to climate change as the things that happen during the spring are changing. The present disjunction is really interesting because we know we have approximately 50,000 individuals in these Western populations and only about 2.2 thousand individuals in these Eastern populations. So there's a big disparity in the number of individuals throughout the state. So this plant was first listed as threatened in 1988 until 2007. In 2008, it was downlisted and listed as rare. This 2016 grant that that funded this work is an effort to try to better understand this distribution. So some of the boots on the ground botanists have thought for a long time that this species is stable in the West and a low priority for conservation efforts. But they also have noticed in the East that it does appear to be more isolated and might be of some concern and a higher conservation priority. So this plant grows on its range edge, and I just want to talk about that a tiny bit here. I know we've talked about it a little bit in the previous two talks, but edge populations are really important because they're often small, isolated, and or fragmented. And they typically have low numbers of individuals that reproduce and contribute to the next generation of offspring, and that's called the effective population size. And because of this, they can have reduced gene flow and higher selfing rates. This might lead us to think that these populations are of more conservation concern. There's this hypothesis called the central marginal hypothesis. It talks about the differences between populations that are in the central part of the range versus the margins. And some of the predictions about populations in the margins are that they would have lower genetic diversity, higher differentiation among populations, and that they might be the results of genetic drift, or more subject to processes like genetic drift, founder events, inbreeding, and other sorts of bottlenecks. And we also think that these could be generally important because they might be tracking colonization events. They might be indicating suitable habitat that could lead range shifts. And they also, if they are genetically different, they might be important to helping us maintain genetic diversity. So I'm going to move into what we actually did. So here's our map showing our sampling. And on the inset map on the upper right, I've got our range map from before. So we sampled 3 populations in the East, marked in blue, and 5 populations in the West, marked in orange. We collected these in 2018 in the very early spring. But for each site, we collected between 10 and 20 individuals, sampled from throughout the population. And our potential hypotheses were that we had either one population, that this is all just one large metapopulation, that each site is its own population, each that is genetically distinct, or we could consider that we had 2 populations, reflecting this east-west disjunction. So methods are very similar to what you've already heard about. I'll talk a tiny bit about things that I think are interesting. So for field work, this is John Koonsman in the field at the Peach Bottom site. We would sample leaves. For Erigenia, we were particularly careful because we wanted to make sure to not damage the plants. So we sampled generally a fragment of a leaf, and we put it in a little coin envelope and put those coin envelopes in a bag with silica gel to quickly dry down the leaves. In the lab, we extracted DNA, and then it was subjected to this GBS process that Cheyenne outlined really nicely. Basically, you cut up the DNA and you sequence all these little chunks of DNA and you get a lot of data. So our goal is to obtain lots of small pieces of DNA and compare them across all of our samples to try to get a genome-wide signal of these little single nucleotide polymorphisms, or SNPs. So our DNA sequences were sorted and assembled using iPirate, as John mentioned. After we assembled the data using iPirate, I filtered it pretty hard to get just SNPs that were present among most of our samples. And this generated a sort of data that looks kind of like this, where we have a bunch of samples and all of our loci in a matrix. And then I used different packages in R to try to estimate a bunch of different things. I'm also going to be showing you a splits tree network, which is just a genetic network that is really fun to look at. So overall, DNA extraction and preparation from Erigenia was a great success. And this is one of the sites. This is also that Peach Bottom site. So this is what it looks like when Erigenia is growing. I used 14,350 DNA sites or SNPs from 118 individuals in our final data set. And so we kicked out only 2 or 3 individuals due to low data quality. And over the next 5 slides, I'll show you some of the results from our 6 types of analyses and try to walk you through these. So the first thing I want to talk about is FST, or fixation index. And this is a measure of the population differentiation due to genetic structure. And the colors of the chart, you can see 0 would indicate panmixis, or that there's no structure, that all of the populations are genetically similar. And 1 is off the charts. It would be like there's no sharing of genetic diversity. And generally, values above 0.25, somewhere down here, indicate very great genetic differentiation. So here on this plot, each population is a column and a row. This is a pairwise comparison. And we can see that there's differentiation between the East and West. So populations in the East, I've marked down here, the West up here, and the comparison between them right here. Overall, we have differentiation, and FST is a bit higher in the East, which is interesting. So I want to talk a little bit of heterozygosity and inbreeding. The first column is our populations, the abbreviation for our populations. The East is in blue, West is in orange. And here it's calculated as an average across loci, and it's a valuable parameter used to estimate the degree of genetic variation within a population. In our third column, So in our third column, we can see that we have an expected value of heterozygosity. And in our second column, we have our actual observed values. Our observed value of heterozygosity is always, for every single population, significantly lower than our expected heterozygosity. And that's statistically significant, as shown by this test, which is Bartlett's K. These values are related to FIS, or the inbreeding coefficient. High FST values imply a considerable degree of differentiation among populations, so we know that our populations are different. But FIS is the proportion of variance in the subpopulation contained in an individual. So high FIS values here, and these are very high, they value from -1 to 1. Values close to 1 indicate high levels of inbreeding within a population. So across the board, our sample populations, inbreeding is very high. So all of our populations have low heterozygosity and very high levels of inbreeding. DAPC, are the discriminant analysis of principal components, is an approach to place our samples in space on a graph to better visualize the spread of our data. It's kind of like a PCA, or principal components analysis, for this data type, and Cheyenne described it much better than I am right now. So if you focus on the points, you can see that each population is a cluster, which I've color coded here. So every population has a different color. Some of those clusters are more similar to each other, which we can tell because they're closer together. And a couple of them, including 2 in the East, Peach Bottom and York Furnace, actually overlap a little bit. But there are some interesting patterns. So the Braddock Trail Park is kind of isolated on its own down here. That's a population in the West. And our third population in the East, Safe Harbor, is kind of isolated down here. But the main thing that I think is really important about this plot is that there's separation along this axis between our West and our East populations. So the subdivision among the West and the East is pretty well characterized by this method. This plot is showing population structure. Here we have each population along the bottom, and they're subdivided by these black lines. And each individual within a population is a bar. And it's important to note that this analysis was run multiple ways, and 2 groups was suggested as the best fitting model for this data set. And so we see 2 colors, blue and orange, which indicates East is blue and West is orange. And so they're pretty well structured and largely distinct. What's interesting is that there is some orange in the blue area and there's some blue in the orange area, and that's suggesting admixture. And that I'm largely attributing to common ancestry. So this is one species that has two distinct genetic clusters. And so this admixture between groups is just showing their relatedness as a species. One of the last things I want to show you are the results of a neighbor net analysis. And so this is a distance-based phylogenetic network. So it's showing the relationship of individuals based on the distance between sequence. And that's really all you need to know about it. What's really cool though is that it's highlighting with this long segment here, the genetic distance between the populations that are in the West versus those that are in the East. And so I thought that was another different and nice way to visualize that. And this was also supported by an AMOVA, which I don't need to talk about, but it's just saying that the variation between sites, over half of our variation in this data set is due to variation between sites. And that value is similar if we calculate it as 2 groups or 8 groups. So tying it all together, our results really do appear to indicate that the disjunction of Erigenia in Pennsylvania has resulted in different genetic differentiation between the East and the West. Overall, FST is very high. This means that our populations are individually genetically distinct. The heterozygosity among all populations across the state is significantly lower than expected, and that is true everywhere, not just in the East. Inbreeding, or FIS, is very high overall. It's values as high as this that are pretty unheard of, in my opinion. And so inbreeding is really prevalent everywhere across the state. A bunch of the plots I showed you are telling us that the East and the West are genetically distinct using different methods, and separation by geography is supported by each of those. And I want to just bring back to the biology of these plants. The biology does also seem to support some of this. So we know a little bit about reproduction, pollination, and seed dispersal, and those are all really important things when we think about genetics and inbreeding. But a lot of that is still not yet known very well for this species, so that might be a future avenue for research. The last thing I want to touch on is that we've really tried to incorporate the knowledge of state botanists here. And we try to work together with them to investigate some of their suspicions. And in doing so, we did end up confirming them. And so here's Rachel Goad and John Koonsman, who I went out in the field with to sample populations in the East. And we just were able to find through this work that all populations are distinctive. They show signs of inbreeding. And in this way, we're kind of supporting that central marginal hypothesis regarding this range edge species. We know that there's been loss of some historical populations, and that extant ones are separated by a ridge and valley by which seeds and pollen are probably not going to cross. We don't have any current evidence for current gene flow between East and West populations. And finally, we know that there are threats that exist to this plant. And those include the damming of rivers. A lot of these populations, especially in the East, occur along rivers, which could bury floodplains. We also know that a lot of these plants occur on privately held land, which is under pressure from development. And also in the West, we sampled from a number of recreational sites. So those could be subject to trampling in the early spring as people get excited about hiking, which is great. So things are really different in the East and West, but this doesn't mean that populations in the West are not vulnerable. So we have supported botanist predictions, but the situation may be worse than previously suspected genetically. We support a reassessment of conservation status in Pennsylvania, and our recommendation is to treat both sides of the Erigenia disjunction as one unit. And perhaps it should be listed as threatened statewide. Certainly it might be rare. And we also sampled a little bit in Maryland, which I didn't show you those results, but the situation is probably similar in Maryland and other range-end states for this species. So I acknowledge our funding sources and people who helped in the field, including Rachel and John. Miley Neal, who's pictured here, is someone who we visited in Maryland to look at specimens from Maryland. I want to acknowledge the place where we did our sequencing, and also my co-authors. So Cheyenne helped a lot with these analyses and getting this kind of analysis pipeline working. Scott Schutte did all of the sampling in the Western populations, and Chris Martin for obtaining funding and his continuous support of these projects. And with that, I would be happy to take any questions.